Have you ever looked at a glass of red wine and just seen, well, wine? What if I told you that you're actually looking at a bustling, microscopic world? Today, we're going to peek behind the curtain and explore the secret science that shapes the flavor, the feel, and even the fate of your favorite bottle. That's right. Every single glass is a living ecosystem, home to millions, maybe even billions of tiny organisms. Now, yeast usually gets all the credit, right? But today, we're focusing on the other influential residents, bacteria. And this brings up a huge question for every winemaker out there. Are these tiny creatures helping them craft a masterpiece? Or are they getting ready to spoil the entire batch? Well, as you're about to see, the answer is a little bit of both. All right, so to really get this dual nature, let's start by meeting the main players in this microscopic drama. Let's meet wine's invisible architects. We're talking about lactic acid bacteria. These little guys are prokaryotic microbes, and they are tiny, about 10 times smaller than their yeast cousins. They can be shaped like little spheres or rods, and they're everywhere. They're on the grapes, they're in the winery, just kind of waiting for their moment. So let's meet the main families, the key characters in this story. You've got Oenococcus, who's often the hero. Then there's Pediococcus and the really diverse Lactobacillus family. Remember those names, because they are at the center of everything that happens next. So, let's start with the good news. How exactly do these bacteria become our friends? Well, it all comes down to one absolutely magical process that can totally transform a wine. And that process is called malolactic fermentation, or just MLF for short. Now, this isn't fermentation like making alcohol. Think of it more like a conversion. The bacteria, mostly our hero, Oenococcus oni, they essentially eat one kind of acid and spit out a completely different one. And that simple change makes all the difference. And this is the perfect way to think about it. The bacteria consume the super sharp, aggressive malic acid, the kind of tartness you get from a green apple, and they convert it into this much softer, creamier lactic acid. That's the same kind of acid you find in dairy, like yogurt. It literally turns a pucker into a smooth, velvety sip. So what's the big payoff? Well, this whole process softens the wine's taste, it lowers the overall acidity, and it creates these amazing new aromatic compounds that add layers of complexity. Think buttery or nutty notes. And as a bonus, once MLF is done, the wine is way more stable, so it's protected from spoiling later. It is a huge win for the winemaker. But what happens when the wrong bacteria get the upper hand, or when they decide to act at the totally wrong time? Okay, now we're gonna flip the coin and look at the dark side, the winemaker's nightmare. Because, you see, if these microbes aren't managed with extreme care, they can absolutely turn on the wine. The very same families of bacteria, just in different conditions, can cause a whole list of really undesirable problems. Okay, here are some of the worst offenders. If bacteria start munching on leftover sugar, they can produce acetic acid, which gives the wine that nasty vinegar taste. If they attack glycerol, they create compounds that make the wine taste incredibly bitter. And, this one's really weird, if they get a hold of sorbic acid, which is a preservative, they can create this unmistakable and pretty awful smell of crushed geranium leaves. And trust me, it gets even weirder. Some bacterial faults create this bizarre, mousy aftertaste that you can only smell after the wine has mixed with your saliva. So strange. Others can make the wine go all thick and oily, a problem called ropiness. Definitely not what you're looking for. So, how do you get all the good stuff without any of the bad? Well, this is where the science of winemaking really becomes an art form. Winemakers have this precise toolkit they use to conduct their little microscopic orchestra. This lays out the main controls, the levers they can pull. pH is huge. Bacteria really prefer less acidic environments. Temperature is also key. There's a sweet spot right around 20 degrees Celsius for the good guys. The alcohol level itself can stop them in their tracks. And finally, there's sulfur dioxide, or SO2. That's the ultimate off switch to stop pretty much all bacterial activity cold. So you see, it's not really about eliminating bacteria altogether. It's about cultivating the right ones at exactly the right time to do a very specific job. It is an incredibly delicate balancing act. And this really gets to the heart of the challenge. Winemaking is basically a microbial succession. It's a race. First, the yeasts have to do their job and convert all the sugar to alcohol. Because if there's any sugar left, it's just a feast for spoilage bacteria. It's only after the yeasts are completely finished that the winemaker can safely give the green light for the malolactic bacteria to start their work.
This quote from our source material just nails it perfectly. It's not that the bacteria themselves are good or bad. The exact same species can be the hero in one barrel and the villain in the one right next to it. It all comes down to the environment. The pH, the temperature, the timing, context is everything.
So the next time you're enjoying a wine that just feels wonderfully soft, round, and complex, take a second. Think about the invisible architects who were hard at work and the tightrope that the winemaker had to walk to make it all happen. It's not just fermented grape juice, is it? It's a masterpiece of controlled microbiology.